using scientific journals as sources count as proof?
What's that?
Would using scientific journals as sources count as proof?
It's a table.
Depends. I mean, you can cite journals, you can lift information out of journals, that's not a problem. But like I said, after reading what you write, you have to convince me beyond a doubt. I can't come up with a counter argument.
So we just have to prove that fats contain more energy than you extract from sugar.
Correct, and you get more energy.
Should be easy enough.
The reason I ask this is because it's always in the text. And like I said, if you look at your text, it really does not tell you how that's really true.
The text doesn't tell you anything.
The text is supposed to tell you
Want the notes?
But as far as this, everybody assumes.
On Monday?
So that's what I want you to do. Don't assume it, and then go back and prove it. It really does.
Whatever.
So that'll be one of your
Brain
quizzes.
is finally all over the place right now.
What's that?
Wait, so you're saying that?
You could disprove it, and you could try. But like I said, what I want is that, you know, all the text saying that,
The 21st.
started last time, that fatty acids are a better source of energy than glucose. I want you to prove it.
Or disprove.
Or disprove. But I mean, really, what you should be thinking of is the energy that you're going to get. So you can make some assumptions. You can make some assumptions that ultimately you're going to use and prove citric acid. So,
Can I copy these after class on a copy?
Yeah.
that's a reasonable assumption. Okay. That as you're metabolizing the sugar and the fats, that it is eventually going to go to citric acid. Okay. So given that assumption, show me that the fatty acids really are better.
And this will be due on that.
How do you do that next time? So you got one.
That's 10 points.
That's 10 points.
Boy. Did you say I'll just drop it?
Could you say a date or
You're going to drop this one? This is a fairly easy one. What's that? I'm not going to set a
You're
date.
just completely setting it already.
I'm like that.
Just make a little bit.
Took me 6 years to get married. What? More than 6.
Could you make
No, I mean, what am I going to do? I'm going to say, I'm going to commit to a date, and then if we're not done the material, you're still going to take it? Right, we're going to try to get done 17 today. So we'll probably get nearly done 18 next week.
Okay.
So, like I said, the soonest that we would have it is the Thursday we come back after break. The latest we'll have it is the Tuesday
Tuesday.
after that. Well, we'll see.
Oh my God, let's be Friday.
We'll
Yeah.
see. Or else everybody comes in on Sunday.
No, I hate coming in on Sunday.
All right.
It's only far.
Any questions before we get started? We're
I
going to
don't like
move
to go in
forward.
for anything at 8 a.m.
Any questions?
Ooh, can you still send
Okay,
me that
everybody's
last
clear on
Yeah.
what's going on?
Yeah, I have
Coming?
like 30 minutes out of 45. My
All
phone loaded right
I'll
now.
put this down again on
It's
the
in-
agenda, but I just wanted
Not the
to
Tuesday that
explain
happened,
this.
just Tuesday the
Because the
12th.
friend will come, the explanation will
Before
be.
the day before the test.
Oh, yeah, I have a recording of it.
I have
But
it
then
here.
also have my own notes. Yeah, I can send you both.
Jackie gave me her notes, I'm going to photocopy those and then I can listen to it.
You
Yeah,
can do
that-
it however you want to do it, but like I said, what I want, you can do it point by point, you can do a
What
comparison,
was
you
the
can do
date?
tables,
February.
diagrams, whatever. But somehow you have to convince me
Yeah,
beyond
David
a
Dropbox.
shadow of
Or,
a doubt. So in other words, you want an
yeah,
argument.
because I store it. on
Really, what you
Dropbox.
should be doing is anticipating any questions somebody has.
Yeah, just send me your email or your Dropbox name and I'll send it to you from there.
For example, if you tell me, oh, well, fat's better because it produces more ATP, I'm just going to load that right out of the way. You're going to get nothing. Pretty much. You get 2 points out of 10 for that. It is 2 points.
ATP's energy, what's wrong with that?
So,
Two.
right. So, give me a very good, like I said, argument. And you're certainly, you can work together, but everybody is responsible for turning their own. Okay.
Everyone can get together.
Okay, so let's continue on that with fatty acids. What's going to happen to them as far as how we make energy? So where we left off was how we were transferring fatty acids into the mitochondria, okay, to get the sort of the energy value out of it. So we said that we had gotten the fatty acid to the cell, and we had made a coenzyme A derivative of the fatty acid. Okay. So then what we had to do was we had the mitochondria. So there's the barrier. Here's our mitochondria. And we got to get it into here. All right. So if the acid's small enough, it can go through, right? But most of the acids are bigger. In fact, most of the fatty acids that you're using are C14 or larger, so they can't just diffuse right into the mitochondria. So they have to get transported. So to do that, we're going to use what? Well, it is a carrier. All right, a carrier based on what molecule? We talked about it. No.
ATP.
Carnitine, right? Carnitine. So again, I'll put that structure up. Not that you have to memorize it, but okay, so that's carnitine. Okay. So what's going to happen is that on this side of the membrane, so here's our cytosol. So we have plenty of coenzyme A on this side. We have coenzyme A on this side. All right. But they're two separate reservoirs. So what's going to happen is right at the membrane, sort of the divide here, what's going to happen is the carnitine is going to react. So it's going to do a transesterification. So we talked about that. We're going to have that alcohol functionality react with the thioester. And energetically, that's very good. All right, because if you remember back in organic chemistry, esters are okay, but this is a high energy molecule. So going from a thioester to an ester, energetically we're going downhill. So that's not a problem. So this is going to attack, right? This comes back, kicks out our coenzyme A. And so as an intermediate, it's going to get transported through right, where this line represents, well, I guess I can draw it out. So we got CH, CH2. Represents that. Okay. So once it gets onto the other side, after getting transported through, then this is going to react with another equivalent of coenzyme A. All right. So again, the sulfur is nucleophilic. It's going to attack. Same basic mechanism. Displaces the electrons initially. This comes back, regenerates the carnitine, which will then get transported back through the membrane this way. So it's this part that's energetic, or I should say energetically demanding. Because this reaction is uphill. All right. But again, so what would drive that reaction? And we saw this on the test question. Even though? Yeah, it's going to be with Chatillon's principle. Okay. So in other words, even though this is an uphill battle to do this, okay, even though we consume, or we will consume this coenzyme A derivative over here, that'll drive the equilibrium forward. Because once we have the coenzyme A derivative of a fatty acid, now we can start doing beta oxidation and start generating acetylcholine A, which is then going to go to the citric acid cycle. So that's how we get it into the mitochondria. What do you think is going to drive the diffusion of the carnitine? What would be your guess? Is it going to be active or passive?
Active,
And if it's active, what's going to drive it?
ATP?
Are you using any ATP on this?
No.
No. So it's passive, right? It's going to be the relative concentrations again of the two. So the carotene is going to be able to go back and forth. Okay? All right. Because actually, the one thing you have to remember, all right, which is a good argument, right, when we're going back to here, how many derivatives of ATP do we need to get this process going? So far, how many? One. You've only used one equivalent of ATP to make this originally. Right? So for every fatty acid molecule, okay, that I'm going to convert to energy, I'm consuming one ATP. Okay. Now, to help you with that, though, that's not a big investment because, right, how many units of acetylcholine A am I going to produce? Well, I'm producing 1 unit for every 2 carbons. So if I'm transporting a C14 fatty acid, right, I'm going to wind up with 7 equivalents of acetylcholine A that's going into the citric acid cycle for a cost of 1 ATP. So the bigger your fatty acid, right, the smaller the initial cost to get it into the mitochondria. So that's why for our assignment, we're going to keep it at C14. A little easier as far as an argument. Okay, so any questions so far? All right. So let's talk about then, once we have it in there now, what's going to happen? How racist? They have what they need. So we try and get that. All right. So the first thing we're going to do soon is we're going to do beta oxidation processes. And beta oxidation processes are going to be used for, well, they're used for everything, but they're going to be used straight through as long as we have an even number, okay, of carbons on the fatty acid. And so as long as the fatty acid comes from, you know, a land animal, that's not an issue, right? Most natural fatty acids are even numbered. Okay. Where do we get the odd numbered ones? No nutritionist in here? Dietitian? They do occur naturally. So you get them in things like nuts, like seeds. Some marine animals, you know, produce high number fatty acids. So we'll talk about what happens to those fatty acids as well. But the majority of fatty acids that are natural, they're even numbered. So we're going to go straight through. Because remember, we're producing acetylcholine A, which is a 2-carbon unit. So as long as we have an even number of carbons, we know that that's all we're going to produce. Acetylcholine. All right, well, let's talk about our reaction. And I think I did give you a handout with those reactions on it. Maybe I did, maybe I didn't. All right, well, it's not, this part's not. So anyway, first thing I want to do is, again, just remind you in organic chemistry how we designate different carbons in a carboxylic acid. So if this is our carboxylic acid, and actually, you know, in this case it's a thioester, but that's originally where the carboxylic acid functionality was. We start designating the carbons as alpha, beta, gamma, so forth, right down the line, delta, as we move down. So when we start to consume the fatty acid, we're actually going to start at the acid, right, the carbon unit. We're going to move this way. All right. So that's the direction of, if you want to call it the degradation or the metabolism, the consumption of this acid. So we're going to start from the functional group. We're going to go down. So the reactions are really easy. There's only 4 reactions. They're very straightforward. All right. First thing that we're going to do, so reaction number 1 is we're going to treat it with a dehydrogenase. So again, you don't have to memorize enzyme names, but the key component that you want to remember in this is that we're using FAD. Okay. Actually, I guess it's just FAD. Okay. So, FAD is a little different, right? That's a flavin base, okay, rather than the NAD. What's the main distinction between the two? Right? There's a couple distinctions you can tell me, but what's the main one mechanistically? All right, so NAD, you know, does what?
It's a transporter.
It is. It's a transporter of electrons. And so how do I get those electrons on the NAD? How do they usually get there? Now, this is exciting because I think I've written it at least 50 times in the course of this course. How do electrons get in there? Exactly. H plus and H minus. So we got H minus, we got H plus, and so that H minus reacts with the NAD. We get NADH. Okay. Plus H plus. Right, that's the typical. All right. And like I said, the book does say, oh, well, you know, okay, we can have 2 electrons plus 2H plus, but, right, this is better. All right. And so if we look at it this way, and the reason why I like looking at this is because it's 2 or nothing, right? You're transporting 2 electrons at a time. Right? That's not the case with FAD, right? You can put 1 electron on at a time. They can go individually. So we have the possibility for radical reactions here where we don't have that possibility with the NAD or the NAD2. So anyway, that's not critical in this particular step, because really it is still just going to act as a hydride acceptor. So what's going to happen here is that, we're going to have a base come in, basically, it's an elimination reaction, where we're going to take 2 hydrogens off the alpha and the beta carbon. Now, what's interesting about this reaction is that it only gives us the trans. So that when we look at this now, this product, so here's our alpha, here's our beta, our gamma. We only get the trans alkene. Now again, you know, just a thought about where we're going. If you have a unsaturated, what's the typical conformation of the alkene if you have an unsaturated fat? Cis. All right. So you know somewhere down the line, if we're going to consume cis, right, what do we have to do to them? Well, based on this reaction, what do you think we're going to have to do then? You do it. All right, you know you need what kind here? All right, so if you have cis, we have to isomerize it, right? We're just going to wind up isomerizing the cis to the trans. All right, so that's a little later. Right now we're just assuming that we got saturated fat. All right, so now we got the trans. Now, why do we need the trans? Well, it's the next step. Because in the next step, step 2, what we're going to do is the equivalent of a Michael addition, organic chemistry. So what is a Michael addition? unsaturated carbonyl. We have two sites of nucleophilic attack. We have the carbonyl, which we know is electrophilic, but also this beta site is electrophilic. Right? The nucleophile can act there as well. So what determines which site you get a reaction at? Back in organic chemistry.
Isn't it like what's more electronegative where?
Not electronegative.
Like-
It has to do with the nucleophiles.
I always thought it had to, wait, isn't it?
They're both electrophiles. Right?
Yes.
We got this and we got this.
Now that we're keeping
It's related to that, but no,
start.
not really.
Doesn't the oxygen have to do with something?
Which oxygen?
The double bonded oxygen right next to the alpha.
Not necessarily, because this holds for whether it's a ketone or an aldehyde or an ester.
The acidic hydrogen.
No.
Does it have anything to do with bitter leaving?
No.
I have no idea.
Has to do with the character of the nucleophilicity, hard, soft, acid-base theory. Okay, we didn't go a lot into that, right? But hard acid-base theory tells us that if we have a nucleophile and an electrophile, they tend to match up. So if the electrophile is hard, meaning it has a lot of positive character, it prefers to react with a nucleophile that has a lot of negative character.
Is it a hard nucleophile?
Hard nucleophile, which means a lot of electron density, reacts with a hard electrophile, which means a lot of positive character. On the other hand, if the nucleophile is not very nucleophilic, meaning it has low electron density, it tends to react with electrophiles that don't have a lot of positive character associated with it. So when we look at this reaction, what we're going to do is we're going to react this with water. Water is my nucleophile. And we know from organic chemistry, is water a strong nucleophile or a weak nucleophile?
Not strong.
It is weak, good. All right, there we go. So we got a weak nucleophile, so therefore the beta carbon is going to be the preferred site of attack. Okay? All right. So again, the water is going to come in. It's going to attack there, right? It forms an intermediate, basically enol, which gets protonated. So the net result is that I now have this. And the critical part of this whole reaction is, now remember, we're not doing this in a beaker like in organic chemistry. We're doing it in the enzyme where there's this microenvironment, induced fit, that's catalyzing the reaction. Only the trans alkene reacts. So if that was a cis, there is no nucleophilic attack. The water doesn't attack it. All right, so that's why there's the necessity to generate that trans alkene.
I'm sorry, could you say that again? Need trans.
Okay, so if we were doing this in a beaker in the lab, it wouldn't matter. Okay, but since we're doing this in an enzyme, where we're dependent on the microenvironment and the whole idea of induced fit, binding and stability, you have a certain orientation in that pocket, all right? Only the trans will react. If you have the cis alkene, no reaction takes place.
What is that molecule called? Last one.
This one? If I had to give it a name, I would say it's a beta hydroxy thioester. So the next thing we're going to do is we're going to take this and we're going to treat it with NAD+. So at this point, you recognize what's going to happen. Something's going to get oxidized. What's going to get oxidized? Exactly. So again, whenever you see this, you know there's an oxidation that's going to take place. Well, this is already oxidized pretty much as far as it can go. So that means we're going to oxidize this alcohol. And so that alcohol, it becomes a ketone. So again, if you're keeping track, right, this is the alpha, the beta, the gamma. So now we're almost there. All right, that was our third step. All right. So our last step now is going to be a retro Claisen condensation, or organic chemistry. All right. So thinking way back when you had organic chemistry, when you see this orientation of a 1,3-dicarbonyl functionality, what you typically think is that, all right, I made an enolate there reacting with some carboxylic acid derivative here. And usually it's 2 esters. All right. So now we're just going to do the reverse reaction. In other words, what's going to happen is that we're going to take coenzyme A, it's going to attack, all right, it's going to generate this intermediate, and if you let me redraw this one, like this, now, when this falls apart, so this pair of electrons is going to come down, this pair of electrons goes over there, this pair of electrons takes that hydrogen, and this just goes back to the sulfur. What I get is now okay, so I have this plus now a new thioester. Right? This now, right, is my beta. Was my beta carbon. This was my alpha carbon. Right? That was the carbonyl. You see this is an enol. We know enols do what? Tautomerize, right? So when this tautomerizes, this gives me acetyl-CoEnzyme A. So what I've done now is I've locked off these 2 units. That's my acetyl-CoEnzyme A. All right?
And you said this was a Claisen condensation?
That's a retro, a reverse of the Claisen condensation. If I reacted this with that to get that, that's a Claisen condensation. I'm doing the reverse of Claisen condensation. So we'll see Claisen condensations when we look at ketone formation.
What are the products again?
All right, so the product is that I now have a new thioester that's 2 carbons shorter, and the acetyl coenzyme A.
But we used an acetyl, we just used a coenzyme A.
Yeah, we just used a coenzyme A. All right. So what's going to happen to this now?
It's going to make a cell acetate.
No, it's in the mitochondria, so what's going to probably happen to it? It could, but more than likely what's going to happen is the phase. Yeah, it's going to go right into the citric acid cycle. Right? So that's how I'm going to generate my energy, right? This is the component that's going into the citric acid cycle. So now, like I said, I've got a thioester coenzyme A derivative that's now 2 carbons shorter. And so each time I go through this beta oxidation, I'm lopping off 2 carbons each time until I get to the last 2 carbons of the original fatty acid. And that goes right into the cycle. So in other words, if I start with my C14 fatty acid, I'm going to generate 7 derivatives of the acetyl coenzyme A. It's going into the citric acid cycle. Does everybody get that?
So you move on to maybe 4 steps. The mechanism.
Well, I mean, come on, you've seen this, right? I mean, at least that one you should know by heart. Okay? That you don't need to know the mechanism. Right? I'm not going to ask you probably the particular mechanism for that. But I mean, you should know what's going on here, like I said. You know, if I give you this and I say, all right, you're going to go to that, you should know that you're going to use either this or something similar to it, like the NADP or the FAD. I'm not asking you to know which exact one you're going to use, but you should be able to recognize what's going on. You know,
No, I'm just going to let you know that the handout that you gave us last time, it makes it very clear.
oh, okay. Better than me, right? Okay.
Can you just explain the difference between FAT and NAD and when you would use either one?
All right, it's not a question of when it's preferred, it's the type of enzyme. Because we never really went into this, I guess, so I can't assume we had this in another biology course. But let's just go back. So both of these are what would be considered cofactors. All right, I'm trying to use the right definitions there. So you know a cofactor is just some species that you know that reacts with the enzyme to react with the species, to push it through. All right. But there's different types of cofactors. So when we talk about organic, we talk about coenzymes. All right. Now, in coenzymes, you can either have the enzyme as a reactant, or it can actually be part of the enzyme itself. The idea is that the coenzyme is always regenerating, but it could be like a reactant. So for example, things like ATP is often, you say, all right, that's a coenzyme, okay, that I need ATP. Well, you generate ATP, and then ATP can make ADP, and then I know that eventually I can convert ADP to ATP. But that ATP is not actually part of the enzyme, all right? So in the difference between these two, okay, the flavins versus the adenosine type, the adenosine are more like the ATP. In other words, they're in there in space, they're in the cytosol, or they're in the mitochondria, but they're more like reagents, like the ATP. All right, so they come, they help the enzyme react, then they dissociate off and they do something else. All right, so they react with oxygen, they make water, generate electrons, they come back to react with the enzyme again. So they're what I would call a recycled reagent. On the other hand, the flavins are usually part of the enzyme itself. They are attached. And so they don't dissociate away. So, for example, when we were looking at, we did see an example, I can't think of where it was. Oh, it was the PhD complex. Okay, when we were looking at the original creation of acetyl coenzyme A from pyruvic, okay, you notice that there was two transporters. There was both the FAD and the NAD present. And so when it was part of the big enzyme complex, the FAD was there, it was attached. And that's where the initial oxidation took place. And then to regenerate that, what we had to do was employ the NAD. So that reacted with that, which then that went off into the, whatever, cytosol, reacted, and then came back. So the main difference structurally is that these are actually part of the enzyme, where this is more like a reagent. It's out there. Okay. Well, like I said, chemically the big difference is that this can behave either as a 2-electron acceptor or a 1-electron acceptor. But this one has no choice. It's going to accept 2 or nothing. Okay.
I also read somewhere that NADH can hold more electrons, or they're a little bit more electron rich than FAD. Or they
The redox
produce
potential is
more-
stronger than the FAD.
Like they produce more ATP than FAD.
They do. Right. So in other words, for each one of these, you're getting, I think it's like 2.5 equivalents of ATP. And with this, you only get 1.5 equivalents. So you need to know that when you answer the proof. Good piece of information there was brought up. All right. So is everybody okay with beta oxidation before we go on and now look at sort of the odd things that we have to deal with? Like I said, the unsaturated acids or the odd number acids. We're okay with beta oxidation?
I have one question. Why does it matter if it's odd or even number of carbons?
Well, because if I have an even number, then I know what I need is the acetylcholine A, which is a 2-carbon unit. So what happens if I have 15 carbons in my acid? So I'm going to get 6 that are 2, generating 2. But when I get to the end, I'm going to have a 3-carbon long unit. So what do you do with it? And that's what we'll learn. Okay, what happens. All right. Reading it out. That leads us, that was a great question, because that leads to the next thing. Yay. So many. That's right. Well, but they're all in your books, so I mean, you can toss them. It's just that while you're in class here, at least you can look at it without, you know. And as Olga says, the handouts are better than my explanation, so, right? That's all right. I like teasing you guys.
Thanks.
All right. Yeah, I mean, I just give you these just so you can follow along in class or make notes on them. That's all. They're in your book. So, all right. So I can erase this stuff. I can erase this too. Is there any- they have what they need? You gotta get these boards redone. Who was it in our organic chemistry class whose dad was the one that redid the boards? Was it, no, it wasn't Connie. It was a guy whose dad refinished these boards and they did the one up in our room when he mentioned it. I know he sat to the left. I can't think of who it was.
I'm glad you remembered.
He'll come in.
I was like, I know, but I'm oblivious. I know, I'm like, I'm not gonna see this.
Here,
Oblivious all that.
all right. So what we're going to do now is we're going to concentrate on what happens at the end. All right, so let's say we start out with this 15-wall unit, right, fatty acid unit that's now going through a series of beta oxidations. So at the end, what we're going to be left with right, is that. Okay. And so now we can't just send that into the citric acid cycle. All right, it's got 1 carbon too many. So what are we going to do with it? So our first one on the diagram, figure 1711. All right, that's going to go through the oxidation. Okay, and what happens to this? So again, these are reactions that you have seen before. But like I said, we'll just go through them quickly. So what we're going to do here in our first step in dealing with this odd number thioester is we're going to do a carboxylation. So we're going to add a carboxylate group to the alpha carbon. Now at this point, even though it says, all right, we got bicarbonate, ATP, and biotin, I hope that you at least have a general idea of what that mechanism is. Okay? Maybe not the specifics, but what's going on here? Why do we need the ATP? What's the role of the bicarbonate? What's the role of the biotin? Well, let's get the easiest one out of the way. What's the role of the bicarbonate? No. It's already oxidized, right? Bicarbonate is this guy. So that's about as oxidized as you can get for carbon. So what's the reason for this? Why is this even in the reaction equation?
It's a carrier for CO2.
Yeah, that's your source of the CO2, right? I want to put this group, right, on the alpha carbon. So where's it going to come from? Well, it's going to come from bicarbonate. Why bicarbonate?
Transport of CO2.
Yeah, right. There's plenty of it around in the cytosol, in the blood, right? Because we don't have a lot of free CO2, okay? It's dissolved in the media. So then we got tons of bicarb. All right, so now the next thing, why ATP? What's the role of the ATP in this? This is a good test question. You can't turn this one in for credit. Quiz. Oh, I didn't mention that too, sorry. We're going to do the same thing, except we're going to not have to do it in groups. Everybody's going to be responsible for submitting a test question, so I'll put that out. One question, but you are going to be assigned a chapter. So since there's going to be 3 chapters, you're either going to be assigned 16, 17, or 18. And so you have to submit a question from the chapter that you're assigned.
Are you allowed to
That
trade
way I
chapters?
get, what's that?
Can you trade chapters with people?
You could trade. If you wish to trade, but like I said, that's how we're going to do it.
Is that going to be first grade?
Yes. There's going to be quiz break as well. So we're going to have tons of quiz breaks. All right. But anyway, so you can't use this question. All right. So we've already decided. We know that this is my source. This carbon is going to be that carbon. Why ATP? What's the ATP going to do for me?
Isn't the ATP be used to take off DOH to make water so that it attacks?
Close. You got most of it, except not the water part. What's the ATP going to do? First of all, what's the ATP going to react with? All right, we got that, we got biotin, and we got bicarbonate. What's the ATP going to react with?
Carboxybiotin.
Carboxybiotin.
That's what it says in this
It could, yeah.
blurb.
Yeah, okay. Well, there's actually 2 ways that you can write the mechanism. And the book is actually presented both, so yeah, I can accept either one. So the 2 ways that you can look at this is that either this reacts with ATP first and makes a phosphate ester, right, an ADP, which then biotin reacts with this. Displaces that because that's now a good leading group, the phosphate. And so now that gives me the bound and of course I'm not drawing the whole biotin. Then this enolate can react with that, displacing this, and then that's how we get the carboxylate on there. All right? Or you can just do it the other way around. So we can switch the thing that, let's see, this carboxylate first reacts with the biotin, undergoes a phosphorylation. No, wait a minute, that would make sense. I don't remember the other mechanism that the book is. I do give the other mechanism as well. But anyway, so you know that the ATP is an activator, all right? If nothing else, you know that it's making a better leading group. All right? So that gets us now our carboxylate group on there at a cost of 1 ATP.
Can you just repeat that one
What's that? Which form?
ATP.
What the ATP is doing?
And the biotin, like the steps.
Right. In other words, the biotin itself is both the ATP and the biotin are creating good leaving groups for the carboxylate. So whichever order that you do it, whether we're looking at it as a phosphate ester, right, or as an activated angle, all right. In either case, all right, I look at it as this way. This is a good leaving group. So we're going to make an enolate out of this, or an enol. So this is going to be my nucleophile, effectively. And this is going to react with that carbon center to displace the biotin. So how we get it on the biotin, right, is by reacting the biotin with a phosphate ester. Right, that's one mechanism.
The carbonate.
So now we got our carboxylate on there. Now this is an odd issue because, again, the enzyme that we use to make this, you'll see that it makes the D, right, malonate. Sorry, D-methylamalonate. So how would we know that it's the D? It's just how you draw it. So in a Fisher projection, it would look like this. That's the D. Like that. The next thing that we're going to do is we're going to do an isomerase. So we're going to convert the D